Hello, everyone. My name is Henry Tee Bahnson. I'm from Benaroya Research Institute. I'm a biostatistician, and I will be discussing the quantitative response metric, or QR, of C-peptide. Here's an overview of my presentation. I'll discuss the statistical properties of C-peptide and how it makes it a precise metric and useful metric to study type 1 diabetes. And the QR, specifically the quantitative response metric, improves the utility of C-peptide primarily because it adjusts for predictable variation in C-peptide decline. And this improves statistical powering and clinical trial feasibility. It also standardizes and personalizes comparisons between observed and expected measures of C-peptide decline. And also allows for identification of responders and non-responders, which is a useful property for adaptive clinical trial design. Here I'm giving an overview of the data presented herein. So we've got 9 different studies from the ITN Immune Tolerance Network and TrialNet with about 800 subjects. And most of the data presented will be focused on the placebo data. And what I'm showing on the right here is just to make the point that these studies, although very similar in their inclusion criteria, do have subjects of different ages and a different baseline C-peptide, which is relevant to the QR metric that I'll be discussing. And I'll also give an example of a reanalysis of the ADATE trial, which is a positive study using a anti-CD3 monoclonal antibody. So briefly, the statistical properties of a good metric generally for studying disease. Of course, you want it to be clinically or scientifically meaningful. Most of the other speakers will discuss this concept and the relevance of C-peptide to type 1 diabetes. But basically, you want a metric that is relevant to the disease state, irrespective of whether the disease is controlled. For instance, glucose levels. Of course, it's unethical to let the type 1 diabetes state to be out of control. So you want an endpoint that really directly measures beta cell function, kind of irrespective of glucose control or HbA1c, et cetera. You also want a measurement that's objective and not subject to a clinician or patient's interpretation. For example, depression scores used for drug approval for psychiatric conditions. We're realizing that many of these drugs don't work much better than placebo, partly because of the real subjective nature of the endpoint. And so the topic that I'll focus on mainly is that the metric is standardizable, predictable. So standardizable, by that I mean rather than comparing to, quote, an average person, a standardized metric allows comparison against some personal expectation. And this is kind of similar to a handicap in sports, where each player is compared to the expectation and put on an even playing field compared to relevant to their ability, their specific characteristics. And it does this primarily by adjusting for age and baseline C-peptide level to give this standardized metric of comparing an observed outcome versus an expected outcome. And then lastly, it's important that the metric is predictable. So once you have controlled, for instance, the baseline value of C-peptide and the age, you have statistical precision that is gained. And this higher level of certainty affords greater statistical power and feasibility. Good qualities for clinical trial research. So on the right, I'm showing the relationship between age at baseline and mean C-peptide values at screening 1 year and 2 years. So these are the same set of placebo subjects looked at cross-sectionally. And of course, you see the relationship between age at screening is positively associated with the mean C-peptide level. But importantly, you see that the difference between the screening time point and the 1 year time point, the drop in C-peptide is much greater in younger subjects compared to the older subjects. So 1 year later, you have a very small decline older and a large decline in people younger. And similarly, between 1 and 2 years, although the fall in C-peptide is less, you still see this relationship where you have a greater decline in younger subjects. And bottom figure just shows the individual subject heterogeneity in the baseline C-peptide value. So of course, this regression line is the same one that's up here, but I'm just showing the individual points from all 300 placebo subjects. And what you see is there's a great deal of heterogeneity, and what the QR metric does is adjust for two things: the baseline C-peptide level, so it adjusts for this variability, and then it also adjusts for age. And so it gives you this expectation of how much, for instance, this subject on the top, who starts out with a very high C-peptide level, what's the expectation of their decline in C-peptide 1 year later? Here's some more information on the actual metric used. Krischer and Bundy published this paper a couple years ago. Here's the equation. I think it's easier to focus on visuals for how the metric is actually derived. So on the left, we have example subject who's 4 years old, and we have in blue the model expectation of what their C-peptide value will be 1 year later. And then at baseline, we have, of course, the real measurement. So this 4-year-old subject had a mean C-peptide value of 0.31 thereabouts. And at 1 year, it declined to 0.1 was the expectation. Whereas what we actually observed with this subject was that they declined to 0.22. So the QR metric is really nothing more than a difference between what was observed and what was expected. So here we have 0.22 minus 0.1 equals 0.12. So, and this is on the log scale. So this subject has a QR metric of 2. So since they did better than expected, on the right, I show that same subject and I compare him to a 5-year-old who had a slightly different trajectory. So again, the blue is the expected, the red is observed. These two subjects started at about the same level, 0.3, but you can see that the 4-year-old did better than expected, so they had a higher QR metric of 0.12 compared to the 5-year-old who had a decline that was about expected and a QR value of negative 0.05. On the right, similarly, you've got a 26-year-old and a 43-year-old. Now they both start and end, about the same value, but the 26-year-old does quite a bit better than the 43-year-old in terms of the QR metric, primarily because their observed and expected value were similar, whereas the 43-year-old was expected to have a decline that was more shallow, and it ended up that the observed decline was steeper, so their QR metric was negative 0.22, whereas the 26-year-old is about 0.6. And in comparison, an 18-year-old subject here, they're important to note that their C-peptide value is actually below both of these subjects on the right, yet the QR metric is higher because, again, they showed that they could retain their C-peptide value from baseline to 1 year, where, of course, this was expected to decline. This slide is meant to show a generic power calculation, simply illustrating the idea that when you have a metric and a model that has a predictable outcome, this affords you greater statistical power, which of course translates to a lower sample size. So in this very simple simulation study or power calculation, I assumed what is often assumed for clinical trials designed in this way. You have a 2-to-1 treatment allocation to treatment to placebo. The mean C-peptide value for the placebo subject is what we observed for the 9 studies shown previously, with a typical standard deviation. And I've assumed a mean treatment effect of it's pretty modest, only 33% better in treatment. So a small treatment effect with about, using an ANCOVA model and 90% power. And then what we see is that for something like the QR, which has an R squared or predictability between 50% and 60%, we need about half the amount of subjects, say around 150, to show a pretty small treatment effect compared to if our metric was not predictable and our R squared value was not as good at 10%, we need about twice as many people. This figure simply shows the kind of natural variability you can expect in QR. So these are all 300 subjects across the 9 studies in the placebo groups. And what you see is that about 50% of the data lies between 0.1 and negative 0.1. So naturally, the top quartile, or top 25%, is above 0.1 and the bottom below 0.1, below negative 0.1. And the point here is that you can define thresholds of quote responders or non-responders based on this distribution of what you would expect to see in a group of subjects if they were not on treatment. So here we're showing, again, all 9 studies on the x-axis. On the top, we're showing the QR metric, which standardizes itself around 0. And on the bottom graph, we're showing the mean decline in C-peptide and its native units. So what you see is across the studies, there's quite a bit of heterogeneity between the decline in C-peptide. But at the top, you can see that the QR metric nicely standardizes this variability. And it standardizes it around an expectation of 0, which is how the QR is defined. In other words, if your QR metric is 0, you have behaved exactly as expected. And so what we see is that on average, these trials do behave as expected around 0. Almost all the confidence intervals overlap with 0, except for one case here for ATG. And this is somewhat expected just by random variability. I mean, there could be something different about the inclusion and exclusion criteria of ATG, but it's likely just due to sampling variability. And this is something that that can happen in clinical trials, especially when you have very small sample sizes. Random variability causes the values to be sometimes different than expected. So in this case, ATG did, the placebos did worse than expected, or its title did better than expected. And these kind of random variations can have consequences for clinical trials because they introduce type 1 and type 2 error when you have this kind of sampling variability. And one positive feature of the QR metric is that in standardized studies, standardized data across many different studies, get a very precise estimate of what the expected mean value of QR is among different subjects with different ages and different baseline values. And what we can see is if you pool all the data from all the studies, the QR metric just nicely sits right on top of 0 and the confidence interval overlaps 0. So in this example, I'm just giving a reanalysis of the ADATE trial using C-peptide at 1 year versus the QR metric. So on the left, we have mean C-peptide at 1 year, and we're just showing value for every subject, placebo versus the treated group. And this was a positive study in that the treatment did significantly improve C-peptide outcome. P-value was about 10 to the minus 3. Now, if you use the QR metric, which again is a little bit more precise, incorporates age, baseline value, it affords you about 2 orders of magnitude on the p-value. So the basic point is that you just get more statistical power and certainty by using a model that adjusts for baseline level and age, which is typically how the analyses are carried out in these studies. So the next couple slides are just a quick disclaimer that this is a somewhat provocative topic. And I really want to ask, are placebos necessary for C-peptide endpoint trials? So given the predictability of C-peptide, is there really equipoise in placebos? We do these studies again and again with placebos, and we see the same thing over and over again. And do we really need sugar pills to estimate what the decline in C-peptide, B, folks are not on treatment. And of course, the argument is that type 1 diabetes is not like depression studies. We have a very objective and predictable outcome, and we really don't need sugar pills to study this decline and compare treated subjects with a group randomized to placebo. And as you probably know, placebos are often not used, for example, in cancer research. In this field is often where you see adaptive clinical trial designs and Bayesian methods that are more innovative and often accepted and common for this therapeutic area. And placebos, of course, can increase speed of screening different therapies and improve recruitment, retention, and cost. So here I'm giving an example of an idea where you just compare a treated group with a synthetic control group. So there's many different ways to do this. Synthetic control groups are kind of a hot topic now. There's also methods where you do Bayesian statistical analyses, or you just compare treated subjects with a historical expectation. So on the left, I'm showing the same data I showed before using the QR metric. And in the middle here, we're comparing against not just the folks who are randomized to placebo in the ADATE trial, but all previous subjects standardized for their age and baseline value, baseline C-peptide value. And what you see is another 2 orders of magnitude significance improvement in the p-value. And you can also think of not using a synthetic control group at all, but just comparing against a historical null value of 0, which we have pretty high level of certainty that this is what the expectation is of C-peptide decline 1 year after diagnosis. So in that sense, you get another couple orders of magnitude by just doing a 1 sample test comparing against a historical null of 0. So this slide really just tries to illustrate that the C-peptide decline is predictable. So on the x-axis, you have the model expected C-peptide value at 1 year after diagnosis, and on the y-axis, the observed. And what you see is the regression line is really right on top of the diagonal. So the mean value closely matches the expected observed are very similar. There are, of course, some heterogeneity. The people above the regression line did better than expected, and those below the regression line did worse than expected. And here you can see an R-squared value of about 0.53. It's important to point out that this QR model was developed on 5 of the studies shown, but then it independently validates in 4 studies. And I think this really supports the use of a historical QR instead of placebo subjects or synthetic control groups or using Bayesian priors, etc. And I want to give just another plug for really the flexibility of the QR metric, which was defined by Krischer and Bundy, using baseline data and projecting forward to 12 months. But you can really use the same kind of concept at many different time points, post diagnosis. So what I'm showing here is a heat map of the R-squared values, which is a measure of the model fit or predictability. And I'm showing the R-squared values for 15 different models. So on the x-axis is the baseline predictor. So for instance, where the subject is at the beginning of the study. Is it right after diagnosis, 3 months, 6 months, etc. And then on the y-axis is some outlook in the future. So not surprisingly, you can see that the predictability is better when you look at a very close time horizon. So just like you have more certainty predicting the weather tomorrow than you do a month from now. As you move farther into the future, here's 2 years, your R-squared value drops to about 0.49 from 0.7. But nevertheless, you do see very good predictability across a wide range of time points. And this is useful for studying drugs or therapies really in a wide variety of intervals after diagnosis, not just 1 year after diagnosis. So an alternative to doing, single arm, no placebo is this trial called sequential parallel comparison designs. And it provides the benefits of a randomized placebo-controlled trial, but uses fewer placebo subjects. And it does this by re-randomizing them into this crossover type of concept. So they start out on placebo, and then they get a chance to be crossed over into a treatment after an interim analysis. And I'm suggesting that the QR metric could be a good metric to use for this interim endpoint to cross over people from placebo to treatment. And this is an article that describes nicely when design should be used. Type 1 diabetes therapeutic area does fit into this paradigm. Here's a brief description of how the study works. Folks are randomized to drug and placebo at the top, and then you have an interim analysis at stage 2, where people are categorized in the placebo group as responders or non-responders. And of course, non-responders would be people whose C-peptide decline, is as expected, and it drops quickly. The idea would be that you re-randomize these subjects to get some probability of getting treatment after being on placebo. So it has this kind of hybrid crossover design built into the randomized study. And this depiction is kind of what we'd like to see. So again, the folks who were randomized to placebo, who are non-responders, they could be identified at 6 months. And then the hope would be that these non-responding subjects, i.e. the people on placebo whose C-peptide decline is happening, would then re-randomize them and gives them some chance of getting treatment. And hopefully you could see the C-peptide decline start to bend away from the projected and flatten out. And so this type of design is a nice compromise between having a placebo group, but then having participants get a greater chance of being exposed to a treatment that may benefit them. So the conclusions here: the QR metric is simple to use. It's a reformulation of a C-peptide, and it adjusts for age at baseline and baseline C-peptide level, and removes a lot of the significant heterogeneity of C-peptide decline. And it provides a nice standardized metric to compare studies, reports results to participants and the scientific community. And it allows for a standardized classification of responders and non-responders, which is useful for single and adaptive trial designs, and lower cost and increase speed of screening therapies, which we greatly need, and give the potential to improve recruitment and retention, and of course, hopefully allow participants of these studies to have a greater chance of benefiting from some of these experimental treatments. Here are the folks that helped with these ideas in the presentation, and also, of course, to Brian Bundy and Jeffrey Krischer for the QR model, and then colleagues here at Benaroya Research Institute, Tolerance Network, also at Trial Design. Thank you for your time.